In our previous video, we have discussed dermatophytosis. In this video, I will be discussing another fungal disease that is Candidiasis, which is also called as Candidiasis and moniliasis. So, in this part of this video, we will be having an introduction in general regarding this Candidiasis. So, here we will be have a look at the causative agents of the disease and how this disease will be manifested in different forms, and what are the different hosts and the diseases associated with those hosts, and what is the habitat of the Candida albicans. So let us start with the causative agent of the disease. Basically, the agents which are responsible for the disease Candidiasis are specifically belongs to genus Candida, and the species Candida albicans. The fungal form of the Candida albicans which is responsible for the disease is yeast form. So please remember, the yeast forms of the fungus is the main culprit for the pathogenesis of this disease. And apart from Candida albicans, there are many other important pathogenic species are there, and they are Candida tropicalis, Candida pseudotropicalis, and Candida parapsilopsis, and Candida cruzi, and in the end, that is Candida rugosa. Now we'll have a discussion on different forms of the disease. As any other fungal disease will be manifested as superficial or systemic, same, this disease also has two forms. In the first form, that is superficial infection, where the infection is limited to skin and mucous membranes of the tissues such as body tissues as mouth, crop, and the genital tract. So this is where the superficial infection is limited, and wherever there will be immune suppressed and immune compromised hosts are there. So in such animals, there is the high tendency of the infection will be of systemic in nature. So in systemic nature, the causative agents will be there, and they start multiplying in bloodstream, and through this bloodstream, they will get a chance to invade deeper into the organs and tissues of the body. So this is about the forms of the disease as superficial and systemic infection. So this is how Candida albicans looks on the Severus dextrose agar, and this is also the colony of the Candida albicans. So now let us have a look on what are the different hosts affected with the disease. In poultry, which include chicken, turkey, pigeon, and ducklings, the main form of disease which is caused by Candida albicans and other species of the Candida genus is crop mycosis. So the most infected part of the poultry is the crop. Crop mucous membrane will get infected through this Candida albicans. So it is also called as avian moniliasis. And in case of swine, a very noteworthy point is that the disease is of a cutaneous form. So this is a very important thing, because in other host we won't able to see the more peculiar cutaneous forms of the Candidiasis. So in swine we will notice cutaneous Candidiasis, and some stomach ulcers are also noticed. And in case of puppies, kittens, cows, and foals, there mycotic stomatitis is noticed, and also enteritis. And in case of mares and bitches, usually there is the genital tract infections are there, specifically the conditions such as olovaginitis and metritis. And in case of dairy cattle, there is mastitis. Actually, this mastitis is caused by Candida albicans, but also species such as Candida tropicalis and Candida pseudotropicalis and Candida rugosa is also responsible for the mastitis. But when it comes to the abortion is generally limited to the species called as Candida parapsilosis. So in case of abortion, there is a major role is played by this agent, that is Candida parapsilosis. But and in case of horses, the pyometra, which is caused by the Candida rugosa. So these are some important causative agent you must remember with respect to examinations. So the things are, in case of horse, if the condition is caused by the Candida rugosa, that is pyometra. And in case of cattle, if it is abortion, then the responsible agent is Candida parapsilosis. And in case of cattle, if the condition is mastitis, and which also involve along with Candida albicans the other agents such as tropicalis, pseudotropicalis, and rugosa. So this is important thing with respect to the important hosts and their associated disease of Candidiasis. Now these are the special notes with respect to Candidiasis, and here is the very well-known term is there, that is thrush. Thrush is nothing but whatever the pathological changes which are associated with this Candidiasis in alimentary tract. So this is in general the Candidiasis of alimentary tract is called as the thrush, and mainly this name has been given in birds. And this is, as we already discussed, in young and even compromised animals are more Candidiasis is more severe, and synonyms such as moniliasis, candidosis, and candidiasis. So this is the aborted fetus and the mastitis images you can see, and this is about how oral and genital tract will get infected with this Candida albicans, and this is the impacted crop, infected crop because of this agent. And now let us have a look on what is the habitat of the Candida albicans. So distribution is worldwide distribution. Basically, these Candida albicans are saprophytic in nature, which means they live on decaying matter and moist environment. And usually they present in the human host only as a commensal. So the commensal is nothing but they normally present in the human or any other hosts. Whenever there will be severe suppression of the immunity of the host, so in such case they will get trigger point and they start invade, and the infection will be carried on. So in normal conditions, these the agents which are belongs to the Candida are commensals, and usually their sites such as they are commensal of oral cavity, gastrointestinal tract, and genital tract. So but in case of birds, a very common site for the this commensals is a crop. In case of birds, the normal site where these Candida albicans and any other agents of the Candida are belongs to are situated or rested in the crop tissue. And when it comes to the transmission, which is mainly occur through the fecal contamination of the feed, water, and environment. So this is about the habitat of the Candida albicans. So now we'll have a discussion on pathogenesis of the Candidiasis. The source of infection involved usually these agents are commensals, so mostly it is endogenous only. The triggering factors such as immune suppression, prolonged antibiotic therapy, malnutrition, and under stress condition will cause the source of infection to cause a severe disease in the animals, which has a large impact on the productivity of the animal. So again, when these organisms are present normally in the host, there is a mechanism of adhesion. So the structures which have play a very important role in adhesion to the host cells are basically mannans and fibrillar peptides. These mannans are nothing but a type of carbohydrate, and these fibrillar peptides are proteins. Both combinedly play a role in sticking to fibronectin of host cells. So these fibronectins are nothing but a glycoprotein, so it's usually present on the cell wall or cell surface of the host damaged cells. So here you can visualize the fibronectin, so which is composed of carbohydrates, proteins, and polysaccharides. So usually this is called as a proteoglycan complex or a glycoprotein. So this fibronectin will be present on the cell surface of damaged cells of the host cell. So this fibronectin will be connected with the mannans and fibrillar peptides. So these mannans and fibrillar peptides are such extensions of the Candida organisms where they will attach with the fibronectin and which facilitate the adhesion to the host cells. Usually in normal conditions where animal is very healthy and which has got a strong immunity, in such conditions these the normal microflora of Candida in the body will be washed away with the secretions such as saliva or any gastric secretions. So whenever the immunity of the animal has got compromised, so in such conditions the organisms of Candida genus will get upper hold and they will adhere to the host cells. Then once they get adhere to the host cells, then they will start invading the tissues. So we already discussed the invasion of the tissue is facilitated by yeast form, usually which is present in the yeast form. So they used to multiply itself and they used to produce multiple different types of Candida through a process called budding. So it is a type of reproduction. So whenever the condition is not favorable for the growth of yeast to multiply itself, so in such condition the yeast will modify or turn itself towards the production of hypha. So these hyphas are elongated structures and they will penetrate the tissues. So thereby there is a damage and tissue invasion is be taking place. So this will be happening in the tissue invasion. So the form of the fungus which is responsible for the tissue invasion is yeast form of the fungus. And another thing is that whenever there is unfavorable condition is there in the body, so in such condition, so the division is blocked. So now it won't produce any other yeast as through the process of budding. Rather, the yeast convert itself into the hypha, and these hyphas are nothing but they are elongated structures. They have got a capacity to penetrate deeper into the tissues. So during that penetration, there will be a damage and tissue invasion will be noticed. So and this process is also supported with the virulence factors such as the cell wall glycoproteins. So usually they have a role like endotoxin like activity, and such enzymes like protease, lipase, and neuraminidase. So these usually damage the host tissues, and other structures such as chitin, mannoproteins, and lipids. They have a larger role in structural and immune evasion role of the host. So this is about how the pathogenesis will be taking place, and in the end the outcome of the pathogenesis is mucosal infection is commonly affected. So the tissues such as mouth, GIT, and genital tract mucosa is mostly involved in this pathogenesis. And another noteworthy point is that in this disease we may not see granulomatous lesions. So this is already we discussed. Immune suppressed animals are prone to the systemic or disseminated candidiosis. And this pathogenesis and pathogenicity will be manifested as lesions and symptoms. So when it comes to the lesions, in the acute case, there will be a small, tiny, yellowish to white or grey pustules are noticed. So they look like a curdled milk stuck on the mucosa of the tissue such as mouth and GIT. And this is a characteristic feature in acute case. And also when it comes to the birds, so in the birds the crop wall the wall of crop will get thickened, and which is again covered by the corrugated membrane. So this looks like a turkey towel. So therefore this lesion in the crop which is affected with the Candida albicans is called as a turkey toweling appearance. So this is very important point. The several examinations have asked me this question. So here you can observe this is how a turkey towel looks. So this is here you can observe the corrugated pseudomembrane which is getting a shape or which is getting an view of similar to the turkey towel appearance. So this is about the turkey towel appearance of crop wall. So again in the previous section only we have discussed the thrush. Thrush is nothing but condition of candidiosis in alimentary tract. So that which usually occurs in the avian species. So therefore it is called as avian moniliasis. So where lesions are mainly noticed in the crop, such again it can spread to the mouth, esophagus, proventriculus, gizzard, and also to the intestine also. And in case of mouth and esophagus, ulcer like patches are also be seen. So this is you can have a look about look at this picture where the bird is dull and depressed and some yellowish flakes are noticed. So these are general points. Poor growth, tough feathers, and loss of appetite will be noticed. And this another important point is that as the crop is get infected, the stance of the bird will be head dropped on its shoulder. Such type of stance will be noticed. And in case of acute cases, there is high mortality seen in the young birds. And in the initial section only we have discussed in swine, we can see the cutaneous form. So these lesions are cutaneous candidiasis, and also the gastric ulcers, vomition, diarrhea, and emaciation are noticed. And in case of cattle, usually those animals which have undergone a prolonged antibiotic therapy, so this antibiotic therapy will leads to the suppression of bacteria. Usually the bacteria will hinder the production and the growth of fungal microflora in the body. So whenever we used a prolonged antibiotic therapy, so in such condition the bacteria population will get diminished in counter to the increased fungal growth. So in such condition, so the cattle will get the disease. So it may also spread to the respiratory enteric symptoms, and the mastitis is the most important symptom will be noticed in this candidiosis. Usually this mastitis is mild form and which is self-limiting, which may often resolve in a week. And in general, these are not so highly diagnostic value for to diagnose the disease. So these symptoms are not much specific. So this is about the pathogenesis and its outcome in the form of lesions and symptoms. Now here we will discuss a very important and the final part of this video, that is the diagnosis of candidiasis. If you have observed this whole video, till now nowhere in this video we have discussed regarding the morphology of the genus Candida. The reason is being, along with the diagnosis of candidiasis, we will discuss the morphology and the characteristics of the genus Candida. So another important thing is that whenever we are discussing about the diagnosis or any morphology of the genus Candida, there are certain terms which used to be very confusing for the students or for any other learner, and those terms such as the yeast, pseudohyphae, hyphae, and terms such as blastospores and the Chlymadospores. So these terms will create a confusion in learner. So better to get in clarity with respect to these terms, and then we'll proceed with the further things. So when it comes to the yeast, so this is how an yeast looks. It is a normal cell which is composed of a cell organelles such as nucleus, mitochondria, nuclear membrane, cell wall, and very important component of the yeast is the bud. So this is a structure which is produced from the reproductive method of the yeast called as budding. Through budding, yeast will produce a bud. In future, this bud will cut off, and again it will become a separate yeast. So that is an young yeast. So through this, so here also you can observe these are yeast, and these are the buds which are on the yeast. And here this is how electro microscopy will look like. So in this image, these all round to oval shaped yeasts are there, and on top of that, through budding, small buds are emerged. So this is about the yeast. Now, from the yeast only, a type of spores are produced. Usually these spores are nothing but they are also a budding cells, and those are called as the blastospores. So here you can see the blastospores. These are nothing but they are a budding cells. Budding cells are called as blastospores. Actually, they take part in the reproduction of the yeast. So therefore, they are called as basic yeast reproductive units. These blastospores are nothing but a reproductive unit of Candida albicans yeast. So in general, they are called as the budding cells, which means to say that these spores are formed by budding from a parent yeast cell. So those structures are called as the blastospores, and they are having a very futuristic characters, such as their size is very small, and they are sometimes oval to round in shape, and they have very thin layer on the spore. So this is what differentiate Chlymadospores. Chlymadospores are resting units. They are also called as resting units or resting spores. The reason is being, these blastospores are found in a favorable condition. Reproduction happens in a favorable condition. So whereas these Chlymadospores are produced in unfavorable condition. Whenever there is unfavorable condition exists for a fungus, it tends to produce the Chlymadospores. They are very large in size, and they have a very thick wall on top of them, and they are resting spores. They are big and round. The term resting spores, which means to say that usually they are present at the terminal end of the pseudohypha. Let us consider this is a pseudohypha. We'll study about what is pseudohypha. Just for time being, just consider this as the pseudohypha. Then on top of the pseudohypha, at the terminal of the pseudohypha, there are a production of the Chlymadospores are noticed. So such type of Chlymadospores are called as terminal Chlymadospores. So, and sometimes what happens, in between two yeast cells or two pseudohyphas, so there is a production of Chlymadospores. So such type of Chlymadospores are called as intercalary Chlymadospores. So this is about their differentiation on the both the Chlymadospores and blastospores. Now another term which is left is pseudohypha. The pseudohyphas are nothing but produced from the buds of yeast only, but they look like a hypha. They are nothing but the chains of elongated yeast cells. So these yeast cells will become elongated, one to one connected. So such type of structure is named as pseudohypha because they look like a hypha. Therefore, it is called as pseudohypha. And very two important criteria are there to differentiate between the pseudohypha and hypha. One is being, in hypha, the cytoplasm continuity is very well exist. When you see the hyphae, here you can see throughout these hyphae, the cytoplasm continuities exist. But that kind of cytoplasm continuity is not seen in pseudohypha because these are a single yeast cells. So they are arranged in chains, giving a structure or giving a appearance as a hypha. So therefore they are not a true hypha, but rather they looks like a hypha. Therefore they call as the pseudohypha. One criteria is in hypha they lack a continued cytoplasm. Cytoplasm continuity is lacked in pseudohypha, but whereas in hypha the cytoplasm continuity is present and which is well evident. And again, in case another factor is that in hypha there filamentous structures are there. The parallelly arranged filamentaceous structures are seen, but such arrangement are not seen in pseudohypha. And another important thing with respect to pseudohypha only is they are usually they have a constriction at the end of the yeast cell, elongated yeast cells, and usually they have a conical end. So they lack a true septa also. Therefore they are called as pseudohypha. And remember, the Chlymadospores and these pseudohyphas will be formed in unfavorable condition. So and another important aspect is that these blastospores, yeasts, hypha, and pseudohypha in genus Candida, they have a switch type of life cycle. Whenever the conditions facilitate for the fungus to being as a yeast, then in such condition the fungus will exist as a yeast form. If the conditions favors the pseudohypha, then in such a situation the yeast will convert itself into pseudohypha, then other morphological structures such as Chlymadospores will be produced. And again the pseudohypha may get transform itself to hypha, and hypha may transfer itself to pseudohypha or yeast also. So this is type of a switch type of life cycle is very evident for genus Candida. So this is very important. You must remember when we are discussing about the morphology and the diagnosis of genus Candida. It is very easy for us to understand all these different diagnostic methods. When it comes to the actual diagnosis, the first method is the collection of samples or specimens. Usually we can go for lesion scrapings, the lesions from the oral, crop or skin tissues. And in case of cattle mastitis, so one can go for milk sample collections. And if somebody wants to go for histopathology, and they can take the tissue biopsy. And again, once the tissues are collected, they will be microscoped. And again, there are very different methods of microscopy are there. Through direct smear, we can observe a slide, or we can go for ground stain, or also we can go for any other histopathological stains. So in direct smear, so again, we can opt 10% KOH method, or we can also go for a lactophenol cotton glue technique. So if in such condition, in the direct smear, we used to get, or we will see that oval budding yeast cell and pseudohypha. So now the things will get more clear, because already we know that what is the yeast cells and what is the pseudohypha. So in direct smear, we will visualize oval budding yeast cells and pseudohypha. And in ground stain, the yeast cell will look like a purple to blue color. And in histopathological stains, many different stains such as phosphamatoxylin can be used. And if want to study the cultural characters of the genus Candida, then there is a need for the culture. So to culture the organisms, a media is required. So the commonly used media is either blood agar can be used or saccharose-dextrose agar. The thing is that the agar should not contain cyclohexamide. The reason is this cyclohexamide are act as antagonists for the growth of fungus. So therefore, it is very important in case of genus Candida that saccharose-dextrose agar should not contain the cyclohexamide. So several times in many examinations this point has been asked. And the incubated, so that media cultured with the Candida should be incubated at the 37 degrees Celsius aerobically up to for 5 days. Then the colony on the saccharose-dextrose agar, they looks like white to cream, shiny and convex shaped are there. So this is how a colony will look like in the saccharose-dextrose agar. And a special media is there for the Candida genus is a BiGGY agar. So this BiGGY agar is nothing but this is the bismuth glucose glycine yeast agar. So which contain bismuth sulfite. So here this is a differentiating media which differentiate the Candida albicans to the Candida tropicalis. Here what happens, the colony of the Candida albicans are smooth, brown colonies are there with no color diffusion in the agar. Whereas in the Candida tropicalis, the colonies are same, they look like same, but there will be a diffuse blackening of the media will be noticed. Here you can observe these two are the colony. This is Candida albicans and Candida tropicalis. So here there is no color diffusion is noticed with respect to Candida albicans, but in case of Candida tropicalis, there is the diffused blackening of the media is evident. So this is about the special media that is called as BiGGY agar, which used to differentiate between the Candida albicans and Candida tropicalis. So remember, these things are very important if you are preparing for any different exams also. And special identification tests are there for the Candida. So these two tests are, one is germ tube test and another is Dalmu's plate technique. So we'll discuss about Dalmu's plate technique before then we'll look at the germ tube test. In Dalmu's plate technique, actually the plate contains agar, and that agar is made up of the cornmeal extract and tween 80. So wholly this agar is called as cornmeal tween 80 agar. So this is very important for one to understand what are the different roles of the ingredients of the agar. So then only we will get a principle behind this technique, and this is how for what purpose the Chlymadospores are demonstrated only on the Dalmu's technique. So this is because earlier we discussed that these Chlymadospores are only formed wherever and whenever there is a unfavorable condition is there for the growth of fungus. So in such condition only the Chlymadospores are emerged. So the media should be such type media, so which used to provide an unfavorable condition for the growth of the fungus. So in turn, the other structures of the fungus will be diminished, whereas the Chlymadospores and morphological structures are more evident to diagnose and identify on the agar. So this is about here. The colony will be inoculated on this agar, and this will be incubated for 30 degrees Celsius at 30 degrees Celsius for 2 to 4 days. So then, based on top of that, a cover slip will be placed. So here you can observe a cover slip will be placed and directly visualized on the microscopy only. The role of this cover place is that actually these Candida are aerobic fermenters. So they require oxygen for their growth of the fungus. But whenever these cover slip is placed on top of the slide, so it will inhibit the presence of air to the fungus, therefore which reduce a favorable condition for the growth of the fungus. So this is why the reason we are using a cover slip on top of the streak of the Candida agent. Again, this cornmeal is contain the corn extract. So usually this corn extract is very low nutrient medium, so which contain nutrient poor conditions. So in such conditions, the fungus growth will be very low to moderate type. There will not be any high growth of fungus will be seen. So wherever this fungus growth is restricted, so in such condition, the morphological structures such as blastospore, pseudohyphae, and Chlymadospores, these will be evidently visible because the other structures of the fungus are diminished. Therefore, whatever the morphological structures such as blastospores, pseudohyphae, Chlymadospores are visible to a large extent. So here, and this twin 80 agar plays a very key role, which usually is a source of oleic acid. Twin 80 agar provide oleic acid. The role of oleic acid is that which helps in the growth and production of Chlymadospores. So therefore, this technique is used to demonstrate Chlymadospores. So therefore, it is specifically used for the diagnostic tool for Candida albicans, because in the Candida, only species which produce Chlymadospores is Candida albicans. So this is very important technique when it comes to the diagnosis of Candida albicans. Then we'll have a discussion on this germ tube test. As the name itself indicate, germ tube. So here you can see this germ tube production in serum, horse serum or any other human serum or any other bovine serum can be used to produce structures such as germ tube. So basically this is a yeast cell. From the yeast cell only, whenever these yeast cells are suspended in the serum and incubated at 37 degrees Celsius for 2 to 3 hours, they start producing a tube. So such tube is called as extended part is called as a germ tube. The thing is that there are actually there are two Candida species are there. The Candida albicans will form a germ tube, but the point is that the germ tube formation doesn't involve any constriction. So which means to say that if this is the yeast cell, then the germ tube will be formed without any constriction between the yeast cell and the tube. This is the cell and this is the tube. So you may not find any constriction in between the cell and tube. So such type of structures, if you noticed in germ tube test, and that will be Candida albicans characters. In contrast, Candida tropicalis involve also the formation of germ tube, but the difference point is that the Candida tropicalis involve the formation of constriction between the germ tube and the yeast cell. So this is very important special identification test for the Candida albicans, which differentiate with the Candida tropicalis. Candida tropicalis involve the formation of and constriction between the yeast cell and the germ tube, but in case of Candida albicans, you will not find any constriction between the yeast cell and the germ tube. So this is a special identification test. Now another test such as biochemical tests can be used. Actually, here also two important things are there. In Candida albicans, they ferment glucose, sucrose, maltose. You may not need to remember all these things, but the only thing you must remember is these Candida albicans do not ferment lactose. So these biochemical characters are asked majority of the times in the exams, and also in case of Candida pseudotropicalis, this shows lactose positive. So this is a very differentiation point between the Candida albicans and Candida pseudotropicalis. Candida albicans do not ferment lactose, whereas Candida pseudotropicalis ferment lactose. And another point is that, so this is already we discussed in the this is Dalmu's plate technique only. Chlymadospore formation is only evidently noticed in Candida albicans. So this is an important point you must remember. And another technique for the diagnosis is use of lab animals such as animal inoculations. Animals such as rabbit and mice will be used for inoculation of the samples. So this is what we have already discussed. So here these are the blastospores. They are very small in size and have thin layer on these cells, and usually they are a reproductive unit of the yeast formed by the budding process. But in case of Chlymadospores, they are usually formed under unfavorable conditions, and they are very large in size, and also very thick outer membrane is there, and they formed at the terminal end of the pseudohypha or sometimes hyphae also. So such type of spores are called as Chlymadospores. So here you can evidently see the intercalary Chlymadospores and terminal Chlymadospores. So this is about the diagnosis. I know this is very long, but there are many important things to understand and learn. Now this is the last part of the video where we will discuss the prevention and the control aspect of the disease. These are very simple things. Just try to correct the predisposing factors, such as do not use prolonged antibiotic therapy, and treat for immune compression, suppression, and malnutrition, stress and poor hygiene, and hygiene and managemental aspect, clean housing, feeding, fecal contaminations. These are general points, and this is very important. Antifungal therapy locally via feed you can give nystatin, so which will be fed orally, used in chickens, turkeys, swine, dogs, and cat. And one can also infuse intramammary to treat Candida mastitis in cattle. And when it comes to systemic therapy, there are mainly two drugs are used: amphotericin B, which act by binding to ergosterol. Ergosterol is present in fungal cell membrane, then which leads to the leakage, in turn leads to the death of the fungal cell. The limitation is that the toxicity is nephrotoxic. So this amphotericin is nephrotoxic. And another antifungal drug is azoles. So they include ketoconazole, itraconazole, clotrimazole, and fluconazole. So they block the ergosterol synthesis. The difference is very evident. Amphotericins, they bind to the existing ergosterol in the fungal cell membrane, whereas azoles will directly inhibit or block the ergosterol synthesis itself. And they are very effective in mucocutaneous candidiasis. And vaccines, there is no effective vaccines are available for candidiasis in veterinary medicine. So thank you.